right, our next speaker is Benjamin Obadia, and he's going to tell us about invasion dynamics in the fly gut microbiome.
Okay, hello. Thank you for the opportunity to present my work. Thank you for being here late to hear about the invasion dynamics in the fly gut microbiome. So flies, as humans, are constantly being challenged by new invaders of their host resident microbiota, but not all invaders are able to stably colonize this environment. So we use the Drosophila as a natural model to study the ecological dynamics of bacterial populations and to understand what are the principles that rule the success of an invasion. So in other words, how the bacteria manage to establish in the fly, to grow there, to interact with each other, and also how do they do to spread via a natural cycle from the host to environment to host again. So we investigated the microbial dynamics of Lactobacillus plantarum, which is probably the most common bacteria found in the microbiota in flies. And for this, we used 3 different isolates that have different ecological backgrounds. So we have one strain that has been isolated from wild flies, one strain from our Cantonese lab fly, and one strain that is from a stock center, and it's a common strain. This one has been isolated from human saliva. So using a capillary feeding system, thanks to the discussions from the Bill Jas lab, we delivered defined dose of these 3 bacteria to germ-free flies. And after a few days, we counted the number of bacteria present in these flies by washing the flies, crushing them, and plating on petri dishes. Also, to avoid the bacteria to grow on the medium and to avoid the possibility that the fly could eat again these bacteria, we transferred the flies every day on a new medium, new and sterile medium. So here is a dose response with the plantarum from wild flies. So on the y-axis are the outputs, so the number of CFU per fly after 3 days, in function of increasing input, the CFU that have been fed to the flies. So the first thing to note, an obvious thing, is that all the flies are colonized after 3 days. The second thing is that the number of bacteria present in these flies is pretty constant, even when as little as 20 bacteria have been fed on day 0. All these points here below 1 represent all zeros, but I spread them so it's easier to know how many flies are behind that point. Interestingly, when we feed the other strains to the fly, the dose response gives a different profile. So the first thing is that a lot of flies are not colonized after 3 days. And the second thing is that the number of bacteria shows a higher variation between individuals. So from this data, we calculated the probability of colonization in a function of the input. And you can clearly see here that the higher the input, the more successful the colonization is, which makes sense if we consider that every bacteria has its own probability to colonize. Having more of these bacteria in the initial population increases the chance that the population will colonize the flies. But you can see that we never reach 100% colonization with these 2 strains, even when we feed millions of bacteria to the flies. So there is something that that constrains the bacteria to settle in those flies. So you might wonder if these bacteria were actually growing in flies, and so we did an estimation of the bacterial growth in vitro, and for that we use the plasmid loss phenomenon. So basically, when a bacteria that carries a plasmid replicates, it has a chance every generation to lose its plasmid. So what we did is to calculate the rate at which these bacteria were losing their plasmid. I forgot to mention that we labeled, obviously, this strain with red fluorescent protein expressing plasmid. So we measured the rates of loss after generation in vitro, and we generated standard curves that correlates the number of bacteria without plasmid to the growth rate. So now we understand better that the low-input wildfly strains, bacteria, have to grow a lot during these 3 days in order to reach the steady state here. And you can see that the higher inputs don't have to grow that much because they already reached this steady state faster. So they just divided enough to be maintained in the flies. However, you can see clearly also that the two other strains have divided a lot, and especially the high inputs have divided as much as the lower input. So in order to explain that, we came with this model where, in addition to the intrinsic probability of bacteria to colonize the flies, represented here by a lottery model in which each bacteria has its own probability to make the whole population win the lottery by colonizing. There is an additional bottleneck effect that reduces the population size and that reduces the chance, that reduces the number of bacteria that can actually win this lottery. So here is just a cartoon of what I just mentioned. One thing we can see also, even though we don't really understand all the aspects of it, is that the bottleneck size can change depending on the strains, the isolates we watch. So if you have the same initial population, you can have a higher input at the end of the time frame. So this becomes more evident when you look at the time course. So here is the number of CFU per fly, and here is the day post-feeding. So you can see, so the initial input for the blue points, the human strain, was 3 million bacteria, so it would be around here. And only a few hours after feeding, there is a dramatic decrease of the bacterial population in those flies. A phenomenon which we doesn't see with the wildfly strain we have. Also, the variability of the human strain on the first days is quite huge, and this could reflect the fact that a lot of bacteria are lost through the bottleneck. And at the same time, some bacteria are trying to divide a lot to catch up and to reach the steady state here. So we next wanted to play with the model by applying different constraints to the fly in order to see if we could change the shape of this curve. And so since it's an immunology session, we tried to look if antimicrobial peptides were activated by the feeding of this human strain of plantarum. So I tested different ones like diptericin, and I didn't find any increase in its expression during this time frame. Also, I just started to feed some germ-free Duox mutant flies, but I don't have enough data to be sure of the conclusion. But since the lab is interested in the microbiota, we fed conventional flies that have their resident microbiota with this strain of plantarum. And what we can see is that the curve is shifted. So first to the right here, and that reflects, that translates the fact that the bacteria have less chance to colonize the gut, which makes sense if you consider that a microbiota is already present, and so bacteria already occupy niches, so it's harder for the invaders to colonize. Also, the curve is shifted up, which reflects a reduction in the strength of the bottleneck. We are not completely sure why is that, but some clues would be the strength of the immune system. So since these flies already have a microbiota, maybe the immune system knows that lactobacillus is not a pathogen, and the germ-free flies have never seen any bacteria, so you feed them any bacteria, they think it's probably harmful. Another explanation would be that the pH the pH would change due to the presence of the microbiota. So we are still investigating that. So in conclusion, and in order to understand what principles rule the success of an invasion, I think our data suggests a system where the interaction between the host, the invader, and the microbiota generates two stable states, colonized versus uncolonized. The barrier between both being the bottleneck. So this study has been done only with one type of bacteria and one type of flies. But you can see already that predicting the outcome of an invasion requires to consider more parameters than just the initial input of these bacteria. So for example, it is clear that the source of the isolates makes a big difference. We tried to explain that by the fact that the bacteria in the wild have to catch the flies really fast because the flies just come and eat on fruit really quickly. So the bacteria has to be adapted to grow faster and quicker in the fly. While in the lab, the flies have been raised on tubes forever, and the bacteria that are associated with them have the opportunity to grow on the medium. So they don't actually really need the flies to be transferred from one fly to another. Where the bacteria are growing is also important. So we are also doing some imaging with our labeled strains of plantarum in order to see where they are growing. And how the bacteria interact with each other are also things that we are trying to assess. Here, please don't ask me any question about electronics, but it's just an analogy with the flies and a circuit where we can know the initial input, we can measure the output, but we don't know really what's happening inside the flies or inside the circuit. And it's really important to know where the bacteria are growing, how they interact with each other, and the local input on defined niche is important to know. Also, it's hard to be able to understand the colonization. And also the resistance, the local resistance from the immune system or the pH, for example, are things that matter. So finally, I would like to thank the Ludington Lab in Berkeley, and thank you for your attention.
Any question?
So are the differences in strain proliferative capacity intrinsic to the strain? Meaning if you took a strain that was fed normal lab food and you put it in a wild type context where they ate regular food, would their ability to proliferate change, or is it diet?
I'm not sure I get your question, sorry.
So I might have missed this, but it looks like depending on the origin of the strain, you had different proliferative capacities for the bacteria when they colonized, correct?
Well, it's not a difference in proliferation. It's really a difference of whether they are able to stick in the fly or not. All the strains we have are growing at the same rate, and actually the human strain is growing faster than the other ones, and that doesn't explain why the bacteria are able to to grow in the flight. I don't know if that was your point.
Well, I guess I didn't understand. I thought that the rate of proliferation is what determined the number of bacteria, but we can talk about it afterwards.
Hi, interesting talk. I wanted to know, do you know if your two Do you know if your two strains are? This is really glitchy.
Maybe.
Do you know if your two strains, your lab strain and your human strain, if they're producing uracil?
Produce uracil? No, we don't know.
Okay. Yeah, because you might be seeing some of your differences based off of like immune system and dual oxidase, and that's been previously shown with lactobacillus. So something to think about.
Yeah, I tried the Duox mutant flies. What I can say now is that the number of bacteria is higher, but I don't know whether they produce uracil or not.
Cool, thank you.
Yeah.
Thank you very much.
Thanks.